Kia ora koutou, and welcome everyone. Thank you for coming along to this evening's talk. For those of you who may not know, my name is Jenny Steen, and I am co-convener with Ilmars Gravis of the Geoeducation Outreach and International Development Special Interest Group of the Geoscience Society of New Zealand. And so we're very excited to have Nic along giving a talk to our group as well, but not just our group tonight. This is actually a talk presented by 3 of our wonderful GSNZ SIGs. So I'd also like to acknowledge Chris Hollis, who's convener of the Paleontology Special Interest Group, and students from the Geoethics Special Interest Group, because one of the wonderful things about tonight's topic, MythBusting the de-extinction debate, is that it's so bridging between all of these disciplines. There's something for everyone, whether you're into fossils, whether you're into geoethics, or whether you're into science communication. So it's going to be a really great session with our wonderful speaker tonight, who is Associate Professor Nic Rawlence from the University of Otago's Zoology Department. And so he'll be presenting his talk, which he has been presenting around the country. It's very popular and is stimulating a lot of debate, and we hope it will present a lot of interesting points for you all tonight. And please put your questions in the chat, and we will have time at the end to go through those with Nic. But yeah, so Nic is really passionate about ancient DNA, paleoecology, a jack of all trades in his own words, but is also a really passionate science communicator, hence his presenting to us tonight. So without further ado, I'd like to hand over to Nic Rawlence to present MythBusting De-extinction. Thanks, Nic.
All right.
Over to you.
Thank you for having me. I'll share my screen. And then hopefully you guys can all see my slides.
Yes, perfect. Thank you.
Right. So thank you for having me. So tonight I'm going to talk about myth-busting de-extinction to sort fact from fiction. And this is really quite topical given Colossal's announcement that, in air quotes, they de-extincted the moa and that they've partnered with Canterbury Museum, specifically Paul Schofield, Kyle Davis, who's a Ngāi Tahu archaeologist, and the Ngāi Tahu Research Centre to attempt to de-extinct the moa. And so I'm not going to need to talk about this much, but we're all aware that throughout geological history, there's been the big 5 mass extinctions, and that we're in another unfolding mass extinction, the 6th extinction now. And it's very evident in New Zealand in that with East Polynesian arrival around about 1250 to 1280 AD, and then with later European arrival, there's been this whole suite of extinctions. With the birds, we're looking at least 80 species of birds that have gone extinct since human arrival. But we've also had various mammals go extinct, lineages of mammals, a couple of plants, a bat, and also a skink have gone extinct as well. So it's understandable why people are interested in de-extinction, of whether people can undo the sins of the past. And so this is really where de-extinction comes in. And a lot of this is also tied around science communication. How do we actually communicate science? And so I suspect for a lot of you, your first interaction with ancient DNA was Jurassic Park. And while we can do a lot of cool things with ancient DNA, we cannot bring back dinosaurs, let them run amok, and no offense to any lawyers or people who are related or married to lawyers, we can't let the dinosaurs eat the lawyers like they do in Jurassic Park. And so this is really where people's perception of de-extinction comes in. But we should really be thinking about Jurassic Park and de-extinction as, rather than bringing back carbon copies of animals that were extinct, as we should be thinking about Jurassic Park and this around genetic engineering. And if you remember that movie, they basically said, well, we sequenced dinosaur DNA and then we filled in the gaps with frog DNA. And that's how the dinosaurs ended up being able to breed, because some frogs can switch sex. So enter Colossal. So Colossal Biosciences was founded in 2021 by Ben Lamb, who's on the left of that photo, a Silicon Valley entrepreneur. And George Church on the right of that photo, who had been working for years around wanting to de-extinct the mammoth, or genetically engineer an Asian elephant to be hairy and cold tolerant. And so They joined forces and launched Colossal, which is now a multi-billion dollar company. And they have de-extinction candidates. So they've got the woolly mammoth they want to bring back. They want to bring back the dodo that was on the island of Mauritius that went extinct a couple of 100 years ago. So the dodo is a giant flightless pigeon. They've teamed up with Andy Pask from the University of Melbourne to bring back the thylacine from de-extinction. And they kept the direwolf very, very quiet. So it wasn't publicly listed as one of their de-extinction candidates, and they were working on sequencing the direwolf genome and the entire genetic blueprint of the animal. And even to all of their collaborators who were working on the direwolf genome research to look at the evolution or the whakapapa of direwolf, didn't know that Colossal had de-extincted the direwolf or made genetically engineered grey wolves to resemble direwolves until it was publicly released. And so very recently, Colossal has announced this collaboration with Canterbury Museum and the Ngāi Tahu Research Centre to de-extinct the moa. And I'll read just some of the quotes. Is Paul Schofield, Colossal's advisor and collaborating scientist, said Colossal are providing New Zealand conservationists with an unprecedented opportunity to create a lost species. Colossal CEO Ben Lamb said that within a decade, a moa hatchling would be a reality. And they've privately said they think this will be about 2 to 3 years. Paul Schofield was 100% positive that this will happen and that it will look exactly like a moa, actually having the real thing out there in the forest. And so a lot of the scientific commentary or pushback, including by myself, has been quite justifiably scathing. So Professor Phil Seddon said the end result will not and cannot be a moa, a unique treasure created through millennia of adaptation and change. And Professor Tammy Steeves from the University of Canterbury said, if it sounds too good to be true, it likely is. And critical commentary from indigenous Maori scientists has been even more scathing that I'll get to soon. So how do scientists do de-extinction? So the only way to de-extinct an animal is through cloning. So this is what occurred with Dolly the sheep. So that's one of those hairy characters in that photo. And so the way cloning works is you take a cell from a donor sheep and you extract its nucleus, so the entire genetic blueprint of that animal, and then you get an egg cell from another sheep and you remove its nucleus, so its genetic blueprint, and you put the donor nucleus and what we call the enucleated egg cell next to each other. You give them an electric shock that forces them to fuse, and then you create this embryo that is implanted into a surrogate. And eventually, after hundreds and hundreds of attempts at this, so with failed embryos or aborted embryos or aborted fetuses, you end up with one Dolly the sheep. Now, Dolly the sheep also died of complications of old age, because if you are going to clone a 40-year-old sheep, the baby sheep that is born is actually going to be 40 years old. It's very much like the curious case of Benjamin Button. But to do cloning, you need really, really high quality DNA. So if you've ever done that experiment where you've extracted DNA from a banana or a broccoli or a strawberry, where you've blended stuff up in a blender, added some cold alcohol and some detergent, and then you get a long skewer and you fish it through and you can pull out these long strings of snot. That's really long pieces of DNA. So like tens of thousands, hundreds of thousands, millions of individual base pairs, A, G, C's, and T's, your genetic code long. And you need this really high quality DNA to do it. But there are, as I said, a lot of complications. So the only extinct animal that has been cloned was an ibex that last of its kind went extinct 3 years prior. And there was some high quality tissue stored in liquid nitrogen. They cloned this individual and it died within a few hours due to complications. And again, took hundreds of attempts to get one clone that died. Now, as I said, you need really high quality DNA. So if we think of high quality DNA as this page from Alice in Wonderland, this is a page out of the chapter Down the Rabbit Hole, which is fairly apt. You can photocopy this page, you can work with this page. Think of this page as the Auckland Yellow Pages, really, really thick, 2 volumes. But if we put that Yellow Pages in a wood-fired pizza oven at 500 degrees overnight, it comes out as crumbs, shards, fragmented, like fragments of the Dead Sea Scroll. It's not high quality. It's like we've gone from the picture on the puzzle box to the individual degraded puzzle pieces. So we can't do cloning with ancient DNA. So how do scientists actually, or colossal scientists, de-extinct an animal? And so what we've got here is a paleoecological reconstruction of North America around about 13,000 years ago. And you've got grey wolves there, the ones that look grey, and the ones that look kind of like with the coat of a dingo or a coyote are your extinct dire wolves. So they went extinct about 13,000 years ago. So what scientists can do is they can sequence all of these little individual pieces of DNA, and they can align them against a scaffold genome or a reference genome. The grey wolf, think of it as the picture on the box, and you can reconstruct that genome. Now, what I mean by that is you can reconstruct the DNA sequence, the genetic code, the A, G, C's, and T's, but it's just the code. You can't reconstruct those long strings of DNA. That DNA is still highly fragmented. And so what they will do is they will compare the DNA of the direwolf to the grey wolf in this example and look for all the bits of the DNA that differ between the direwolf and the grey wolf that make a direwolf the way it is. And that could be genes for coat color, genes for baculum size or penis bone size, genes for body proportion, hair length, bone size, for example. And so what you need to do to basically genetically engineer a grey wolf to have direwolf characteristics is you need to take some really high quality DNA out of a grey wolf, or in this example, what we've got here is we're talking about woolly mammoths versus Asian elephants. It's the same process. So you take really high quality DNA from one of these donor animals like an Asian elephant or a grey wolf, and you can then splice into those genomes synthetic pieces of DNA that match your extinct creature, like a woolly mammoth or a direwolf, and to create this genetically engineered individual. And you now take this genetically engineered genome and you put it in a donor cell, give an electric shock, and it'll start developing into an embryo. Now, how do you actually do that genetic splicing? And this is where discoveries in the past decade have been made, where the discovery of this enzyme called CRISPR-Cas9, think of it as a pair of molecular or enzymatic scissors that you can go to your genome. It can very specifically and very targeted, say, chop this gene in half, and you can delete part of this gene, or you can insert, say, a piece of synthetic DNA that matches your extinct creature. And so what Colossal at the moment has been able to do is probably about 500 individual changes at the same time. And what they did with genetically engineering the grey wolf to match, not match a direwolf, but to give it direwolf characteristics, is they did about 20 edits to 14 individual genes. Now, once you've got your genetically engineered genome and you've turned it into an embryo, you need to put it into a surrogate. So with the direwolf, these embryos were put into domestic dog surrogates and were brought to term, and then C-sections were performed, and the 3 direwolf pups. Colosso hasn't said how many hundreds of attempts, like with Dolly or the ibex, occurred to get those 3 pups. But with some species, you don't have good surrogates. So there's not a good surrogate for bringing back the thylacine, the Tasmanian tiger, or an Asian elephant. So artificial womb technology, like what you've got on the left, will need to be developed. Now, with birds, you can't do cloning. There's the whole chicken and egg problem. So what Colossal and scientists need to do is they actually need to take these genetically engineered genomes and put them in what we call primordial germ cells. Now, that's a fancy term for the cells in your body that develop into sperm and egg. And if they can put these genetically engineered genomes into these germ cells, you can get sperm and egg develop, and then they can perform fertilization. And in the case of moa, they will have to create an artificial egg. So Colossal wants to de-extinct the giant moa, and what you've got here is the giant moa egg in the background and an emu egg in the foreground. So there's no living bird alive today with an egg big enough to act as a surrogate for a giant moa. So you can see there's all of these technological hurdles that need to be overcome. Now, assuming you get through all of those, you have these genetically engineered animals are born. What you've got here is two of the direwolf pups. You've got Khaleesi not on this photo, and you've got Romulus and Remus. Now, they are sitting on a prop of the Iron Throne. That Iron Throne is actually owned by Peter Jackson, who was lent it to Colossal for this photo shoot. And what Colossal is arguing is rather than these being actually the de-extinct animals, they're functional de-extinction. So if it looks like an extinct animal, what they think an extinct animal should look like, it is the extinct animal. Now, interestingly, genetically engineering these genes can have, sorry for the pun, dire consequences. And that's what happened when Colossal tried to change the grey wolf gene for pale coat color to match the dire wolf. It had unintended consequences. So they went and genetically altered 3 other genes. Now, that didn't actually produce the coat color that we think direwolves had. It matches the coat color for what we know direwolves are in pop culture or in the Game of Thrones series, these very white direwolves. So you can tend to think of functional de-extinction rather than saying if it walks like a duck, quacks like a duck, and looks like a duck, it actually is a genetically modified flounder. So just because you may look like what a scientist thinks the extinct species look like doesn't mean you are that extinct species. You need to be able to function like that extinct species in the ecosystem. So what Colossal and these de-extinction companies would like you to think, and Colossal's not the only company, is that de-extinction is serving a conservation purpose. Now, with Colossal announcement with the moa is that what they're going to do is they are going to ship artificial moa eggs containing giant moa chicks back into New Zealand. They will be hatched in Ngāi Tahu land around Glenorchy, and they'll be raised parentless. Now, the only ratites that get raised parentless are when we take kiwi eggs out of the wild, drastic measures for drastic times, to rear them in captivity until they are a year old and they can fend off predators. All ratites in the wild have parental care. So there's going to be no moa parents to teach these genetically modified emus how to act like a moa. But Colossal like to say they're filling a conservation need. So you release mammoths back into the wild, they trample the Siberian steppe, sequester carbon, and turn that Siberian environment back into the very productive mammoth steppe. And with the New Zealand forest, it hasn't actually realized moa have gone extinct. There are a whole lot of ecological services that moa perform, like whether that's spreading ectomycorrhizal fungus, these very colorful truffle-like fungi, so your blues, your reds, your purples, that have got a symbiotic relationship with New Zealand forests that help them spread, say, with changing climatic conditions from cold glacial periods to warm interglacial periods. And then you've got plants that have anti-moa browsing defenses, whether it's divarication or whether you've got heteroblasty, so the classic juvenile lancewood versus an adult lancewood. But you're only going to get those ecological and conservation effects if you actually release large populations of moa out into the wild. So what Ngāi Tahu and Colossal and Canterbury Museum want to do with having effectively a moa park for ecotourism purposes is not conservation. It's money making. So there are a whole lot of technological limits around. And so one of them is actually looking at genetic differences and how much genetic engineering you need to do. Now, the grey wolf and the dire wolf share a common ancestor about within the past 2 to 6 million years. Now, 99.9% of their DNA is identical, but the bits that are different are still significantly over 1 million individual letters of code. So doing 20 gene edits to 14 genes does not make a direwolf. And one of the critics really nicely said, to put it in context, is you have to go back 42 million years to get to the common ancestor of a human and a howler monkey. Genetically engineering a howler monkey does not make a human. If we think of the thylacine and its closest living relative, the cute little fat-tailed dunnart they're tens of millions of years divergent. So there's going to be even more changes that need to occur. And with moa, if we think of the closest, the common ancestor of moa and tinamou, that's 58 million years. And then to go back to the common ancestor of moa and emu, you're at 65 million years at the time of the dinosaurs. Now, that is around about 10 million individual DNA differences that Colossal want to do. And they said they want to take a moa genome and change every single individual letter of code to match a moa genome. Now, that is a huge amount, and the technology is not there yet. Now, it's also not just about genes. So the gene for wing formation or limb formation in moa is completely functional, even though moa are the only flightless bird that actually do not have any wings at all. So you could put that gene, moa gene for limb formation, in a mouse model or a chicken model, and those embryos would develop wings. What has occurred in ratites, the family that moa belong to, is it's actually all the regions upstream of that gene for limb formation, what we used to call junk DNA, that are your regulators, your promoters, enhancers that control gene expression. And that's where all the mutations are. So it's not just the genes that are important, it's the other parts of the DNA. And the analogy is that the planes to build a moa are like the planes to build a Volkswagen in the factory. They're completely functional, but it's the computer code that tells the robots how to build that Volkswagen is what's corrupted. And we've also got to take into account that genomes are packaged in different ways. There's different structural variants, different ways there's packaged. There's methylation, so genes being turned on and off in different ways. That just genetically engineering an emu or a tinamou to look like a moa does not make a moa. And the bird that will result will bear no genealogical or whakapapa resemblance to the moa that were here when Polynesians arrived. So there are also ecological concerns. While the giant moa is a generalist, so all of the reconstruction of the diet we've done with isotopes, with coprolites, shows it's a generalist and it could survive from one end of the country to the other and from sea level to alpine. Colossal wants to de-extinct all of the moa, and some moa have very strict habitat preferences. If we can think of like the heavy-footed moa, which is like the cartman out of South Park, or a 40-gallon drum walking on little kids' gumboots, loved edge habitats. We can think of Cresta moa was literally in the northwest South Island, and there are moa species like Easter moa that's across the eastern South Island and down into Southland. Now, that open dryland, shrubland, grassland forest mosaic is very different to the forest we think of in New Zealand. It's more similar to like open eucalypt shrubland in Australia. And there's no analogue left of that in New Zealand. And if we drill down into central Otago, a lot of the work of Mike Pohl has shown that the canopy of central Otago was lancewood and kowhai. It's strange to think of a forest like that. So you can't bring back the species unless you're going to bring back the habitat. And it's like if we go back to Jurassic Park again, the Triceratops gets sick in the first Jurassic Park movie because it's eating a plant that wasn't around when it lived 65 million years ago. So you can't have an extinct species without having the habitat. Now, importantly, bringing back an extinct species is one thing, or even a poor facsimile is one thing. You should really be de-extincting its gut microflora and its parasites as well, because it can't function in the ecosystem without its gut microflora and parasites. Now, we know moa spread the spores of these truffle-like fungi, and we know these spores can go through the guts of, say, introduced animals like deer, but they do not germinate. The exotic fungi go through the guts of deer, germinate, and that helps spread wildling pines. So how much of this ability for these, say, spores of fungi to pass through moa guts and help forest spread is actually down to their parasites and nectar mycorrhizal fungi. And it comes down to is you can't have a genetically engineered animal performing its role in the ecosystem without all of its internal biota as well. But there are also other ecological concerns in that there was a writer by a science writer based in Dunedin who writes in the American magazine Slate magazine. He's got the unfortunate name of Eric Trump, no relation, who said this moa will be a new creature, not an animal with native roots in New Zealand. This makes it yet another introduced species like the rat or the stoat. And so this, say, frankenmoa, it could be really hyper adapted to the changed conditions in New Zealand, and so might require culling. And so it's not seen yet whether tangata whenua will actually treat these moa as taonga or treasured species. So you're going to have all of these conversations around actually having to cull treasured species. On the flip side, and the very real possibility, is it'll be maladapted to the conditions and require a lot of conservation. And so while the money to de-extinct animals and develop this technology comes from the private sector, philanthropists, gullible movie stars and celebrities, the money to actually conserve these animals once they have been de-extincted actually will fall not to Colossal, but to governments and Department of Conservation and iwi. And so we can think of that if a moa got de-extincted and the money to conserve it fell on the government, then it's going to take money away from other species that are more worthy of conservation. If the conservation in perpetuity fell to Colossal, then you're going to have more money to conserve other species. And if you actually just didn't bring back a poor copy of a moa, then you'd actually have more money to save the species that are actually more worthy of conservation. Now, if you are going to do de-extinction for conservation, as I was saying before, is you need to have a large number of individuals. Now, this is to ensure that you are not genetically inbred, like this guy here. This is one of the Habsburgs with the classic Habsburg jaw. They got so inbred that they couldn't have children. Or like, if we think of the English royal family and Queen Victoria being the grandmother of Europe and haemophilia spreading throughout the royal families, or Tutankhamun's children that died very young because their parents were brother and sister. So you don't want individuals to be inbred. You want them to be a genetically variable population. And so in conservation biology, there's this rule called the 500 rule, where you need hundreds of individuals to be genetically secure. And you need the genetic variation of my listeners here or of your university class. Everyone looks different. And this becomes really important if we think of actual mammoths. So mammoths went extinct on the mainland of North America and Eurasia about 12 to 13,000 years ago, and they survived on a couple of islands. One, St. Paul, around here, where they died out due to poisoning of their water source from salt, and then one on Wrangel Island, where they became dwarf mammoths. Now, scientists have actually reconstructed the demographic history of these mammoths, and they've actually looked at ones on the mainland and ones on Wrangel Island. And so if you look at the genomes of the ones on the mainland, they've got lots of heterozygosity, so they've got a lot of genetic variation, and they've got what we call small runs of homozygosity. So in your genetic blueprint, in your nuclear genome, you get one copy from your mum, one copy from your dad, and when your cells divide or when sperm and egg come together, the DNA crosses over and it goes through recombination. And so if you've got large population sizes, you've got random mating, the bits you inherit from your relatives through time will get smaller and smaller. And so that means there's lots of random mating, large population sizes. Now, Wrangel Island mammoths, as they got isolated on the island with increasing sea level rise, went through a massive population bottleneck down to about 10 individuals, and then recovered to several hundred individuals, a stable population until their extinction. And so in conservation, you say that population is recovered, job done, it's saved, it's genetically secure. But that population had a lot lower levels of genetic variation compared to the mainland, and it had these longer runs of homozygosity, so longer stretches of DNA, the copy from your mum, the copy from your dad, that are identical. So that means there's small population size. It's non-random mating. It's mating between close relatives. So you need a large population size of genetically diverse individuals. Otherwise, you're going to have these ticking genetic time bombs and these ghosts of bottlenecks past. So with de-extinction in Aotearoa, there's been a lot of talk. Colossal's announced they want to de-extinct the moa. There's been talk about Haast eagle. Haast eagle could be interesting because if that ever got brought back, you would actually need eagle insurance for tramping trips in the Southern Alps, much like kangaroo insurance in Australia. Lots of talk about the huia. But with New Zealand, with the thylacine in Tasmania, Colossal's efforts to de-extinct animals in the Pacific or the direwolf in North America, there are indigenous people that in their viewpoints are critical to this. And so the pushback from indigenous scientists has been really, really scathing with us. And so I'll just read you one quote. Aroha Mead stated that de-extinction is a misnomer, a false promise that is rooted more in ego than in a genuine effort to conserve species. These are exercises in the egotistical delight in the theatrical production of discovery devoid of ethical, environmental, and cultural considerations. And so we need to be able to talk, and the engagement has to be meaningful. It has to be in good faith. It's got to be ethical, and so that iwi, hapu, runanga, and trust can actually make free, prior, and informed consent based on all the information. And so for Maori, and actually even Aboriginal Australians, Polynesians, Native Americans, as whakapapa is vitally, vitally important. So these indigenous cultures can trace their ancestry back to all living in extinct flora and fauna. And you do not mess with whakapapa. Messing with whakapapa is viewed with a whole lot of disgust. And so that is why these Frankenmoa could actually for example, not be viewed as taonga species. And as Phil Wilcox, who's an indigenous Maori genomics researcher that I've written pieces on around indigenous concerns to de-extinction, has said it remains to be seen if these artificial facsimiles of moa that do not have a whakapapa relationship with Maori will be accepted as treasured. And so this is where I can say that Colossal actually approached our lab and said, we will pay for all of your moa genome sequencing, tens of thousands of dollars, so long as we can use it for de-extinction, and so long as you give us all of your iwi, hapu, runanga, and trust contacts. And we have been, our research group has actually been sequencing moa genomes to look at their evolution and sequencing the genomes of other living extinct birds. And we've engaged widely across the motu, across the country, including with several Ngāi Tahu runanga, with all of the 8 iwi at the top of Te Ihu in the South Island and across the North Island. And I can say with all the people that we have engaged with, there is no support for de-extinction at all. So we were quite happy to tell Colossal, no, we will not become your enablers. We have ethics. So it actually really calls into question one of the quotes Paul Schofield said on the TV3 News the day the moa got announced, where he said there's a lot of jealousy out there in the scientific community that they're not involved in this project. And it's like, we're not jealous. We have ethics. And so when the moa de-extinction stuff got announced, there was a big glossy video, big press release, and it was announced that that they were doing this in partnership with the Ngāi Tahu Research Centre, and they were doing this for all of Ngāi Tahu and all of the South Island. And I can confidently, given all the engagement that we have done and stand on solid ground as a Pākehā researcher, I can confidently call bullshit on that. And so with that and the kōrero, the conversations we've actually had, is Colossal went to Canterbury Museum and Paul Schofield. Paul went and talked to Kyle Davis, who's a Ngāi Tahu archaeologist from Runanga down in Murihiku, down in Southland, whose Runanga are all against de-extinction. And they went, now Kyle used to work for Paul, so there is a conflict of interest there. And they went and talked to the Ngāi Tahu Research Centre, which is an independent research centre at University of Canterbury. And the conversations were had with Mike Stevens, the head of the centre, who's an historian, Tamari Tau, who is an historian there, and who's the upoko, or the head of the local Runanga in Christchurch, Ngāi Hariri. And that is where engagement stopped. There was little to no engagement with head office at Ngāi Tahu. There was little to no engagement with Ngāi Hariri, and there was no engagement with any other Ngāi Tahu Runanga or any other iwi in New Zealand. And importantly, any of the 8 iwi at the top of Te Ihu, the South Island. So the engagement is very much smacks of cherry picking, colonialism, but also it's what Colossal is doing. We've been talking with the Palawa Aboriginal community in Tasmania, who are the kaitiaki or guardians of the Tasmanian tiger, and they were approached by Colossal, and they said, look, this is one of the darkest things you've asked us to do. We are not interested in this. The answer is no. And Colossal didn't take no for an answer, so they went and cherry-picked 3 people who do not represent the community, and 2 who are new to the island, and said, we've got full engagement and support. And so there are many concerns with Maori around de-extinction, with all the ones that I've talked about, also around by prospecting, monetization of indigenous data, sovereignty, indigenous IP, trademarking, but also this breaks Article 2 of the Treaty of Waitangi that Maori have rangatiratanga, or sovereignty over taonga treasured species, and the Y262 claim that Maori can claim intellectual property rights over New Zealand flora and fauna. And so this is all linked in with discussions around lifting the ban on genetic engineering that Maori have been shut out of. And so it really comes down how you actually communicate. And so Andy Pask is actually on record. He's the scientist in charge of the thylacine de-extinction, a public talk, where he said it is hard enough communicating or telling educated scientists what we do, let alone the general public and indigenous peoples. So if you are talking to the public about de-extinction, they're going to have a very different perception than if you say I'm going to go genetically mess up an emu genome to create a poor copy of a moa. And so Helen Taylor, who used to be at Otago University, did a study looking at views around this with conservation practitioners at DOC in 2017. And there was more support for using this technology, this genetic engineering technology, to deal with pests and then save the species we've got left, and more so than bringing extinct species back. And so really what we should be doing is, rather than bringing back poor copies of extinct species just to assuage human guilt under some misguided duty to undo the sins of the past, is we should actually be using this technology and developing it to save what we've got left. And again, this is going to take proper good faith engagement with indigenous peoples, but we could reintroduce lost genetic variation into our endangered species that gives them back that adaptive potential in the face of disease and climate change in a fast changing world. We could make them resistant to disease, like make kakapo resistant to lung fungus or crusty bum. Colossal has done a single base pair change to betongs that make them resistant to cane toads. But we could also be using this to develop gene drives or make sterile Trojan males of predators to introduce into the population to eventually cause population collapse. But you don't need to develop these technologies for conservation. You don't actually need to develop them in extinct species. You can develop these in living species, and you don't need to do this whole de-extinction angle. And so this is really where it comes down to science communication. And when it, as I've been hinting at, is when it comes to Colossal and all the science communication, it's never let the truth get into the way in the way of a good story. So rather than talking about genetic engineering, they're talking about de-extinction. They're very much like a used car salesman. They're selling the promise, the dream, and they are very much working off if we think of scientists, when we put an embargo on a paper, it gets sent out to science journalists, the New Zealand Science Media Centre. They go talk to other scientists. They get critical commentary, see what other scientists think in that. And this is very much very different to the way they do it, where there's these embargoes on what Colossal wants to do. It gets sent out to journalists that are going to write positive puff pieces about it, and they really don't want this scientific criticism or these chinks in the armor of the promise, because they need to be able to convince the next tech bro or entrepreneur or celebrity or private funder to give them hundreds of millions of dollars. And so one of the actual scientific advisors for Colossal, Tom Gilbert, who's a paleogenomics expert, has actually said, well, a lot of this pushback from scientists is actually of their own making, because if Colossal, their communication arm, keeps potting out, saying they're going to de-extinct a mammoth and keeps potting out pictures of mammoth, then when all the pushback comes, what do they actually expect? Because the public perception is actually going to be you're going to de-extinct a mammoth, not genetically engineer a hairy Asian elephant. And so Colossal hasn't liked this. So there's been very few of us actually speaking out. There's a lot of paleontologists speaking out around the world, but very few ancient DNA researchers. And part of that is a lot of ancient DNA researchers will be scared because Colossal's a multi-billion-dollar company, have an army of lawyers. But if you're a junior researcher in an ancient DNA lab, the head of your lab is either friends with scientists at Colossal, because a lot of them are from the ancient DNA world. They've collaborated with those scientists, or still are, or they're in bed with Colossal on these projects. And so there's been a lot of pushback from Colossal with all the scientific commentary we've been giving as our critic and conscience role that's enshrined in legislation. And so Ben Lamb's the CEO, and he's tweeted out, "Wolves don't lose sleep over the opinions of sheep." And then when I wrote a conversation piece with a very tongue-in-cheek title saying real de-extinction is unlikely to fly, he said there are sometimes crazy, weird conspiracy theories about Colossal which make us laugh. But the dumbest headline of all time goes to this article whose author doesn't even know moa couldn't fly. If moas fly, we really effed up, lol. I wish people did more research. And so it's clear he didn't actually read the article where it clearly said moa couldn't fly. So they've got this very thinned skin. And what the critics have also received, so not just me, but other scientific critics, so we have no idea whether these are coming from Colossal or the PR companies they've hired or their supporters. If you start getting these AI-generated hit pieces that basically try to undermine the scientific commentary that you are giving. So this is very similar to how a lot of the people giving scientific commentary in the COVID pandemic were being undermined by anti-vaxxers or anti-government people. And so what we've got here is this one of them saying that I care more about self-promotion than actual going and improving my mediocre publication record. The bottom one is saying that I'm involved in a double standard, that I use ancient DNA to reconstruct past environments. So how dare I criticize Colossal that uses ancient DNA? And you can brush those off, but they start getting very personal. So some pieces against other critics have said the top scientists at Colossal are female. You are criticizing Colossal. You are a white male. Therefore, you are a misogynist, and you are now joined the ranks of famous misogynists through time and a stamping down on top female researchers. These ones, including one that came out just a while ago, are basically saying that a lot of my commentary, despite all of the engagement we have done across the motu, misrepresents Maori perspectives, and that I'm morphing all Maori, iwi, hapu, Runanga, and trusts into one entity. And I can confidently push back on all of these given the engagement we have done. But clearly, Colossal or its supporters do not like being called out for doing cherry-picking engagement in science in the journal. And the Palawai community actually had a really even more stinging word for it, as they called it black cladding. So cladding your project in a veneer of indigenous support. But with all of this, with science communication around a lot of these controversial topics, it's important to push back. And that's what we have been doing, whether that's interviews for Nature or New Scientist. And it's actually really telling. They interviewed Ben Lamb, the CEO, in these pieces, and he says, we want to inspire scientists, not to basically punch them down or tear them down, and they want to run towards critics, not away from them. Yet the exact behavior of Ben Lamb in the tweets, for example, that he's been putting out, the social media by Colossal and its supporters is the exact very much opposite. And so the pushback can come also in the form of media articles, the ODT, including one that actually came out today, calling out these anonymous pieces. And it goes back and forth. So Colossal doesn't like the pushback that they're calling people who are sticking up for the critics nerds that hate us, and very much live in an echo chamber. So Ben Lamb has actually said on Twitter that he thanks Elon Musk for fixing Twitter and making it fun again, and has actually put out these memes saying, basically, if you look out to the sky, all the positivity Colossal is receiving in Twitter. And Simba's asking, what is that shadowy place over there? And that's blue sky. And that's where all the scientists, when Twitter started going downhill, left to blue sky. And then, interestingly, PR companies purporting to actually work for Colossal are sending AI-generated pieces to well-known science journalists like Laurie Winkless, asking them to publish these pieces under their own name and saying that the scientific community are leading coordinated attacks against Colossal, who is the next hope for the next generation. But importantly, with all of this, is I'm actually involved in a project where we're going to start through surveys, is actually collecting baseline data on what people's perception of de-extinction and genetic engineering is, and how that changes with different forms of science communication. And longitudinally actually track this. So whether we can talk about popular science articles or conversation pieces or public talks like I'm giving here, or even satirical tongue-in-cheek cartoons that were done in the ODT over a series of weekends of ways of science communication. How can that change people's perception? And I especially like the second one, where the unintended consequence of bringing back a Morris that has teeth, and then it eats Peter Jackson and the scientists, and then basically they cut the feed so it's not embarrassing. And then this one's talking about what actually happens if the animals escape. And right through to the ODT, each week actually has a section on teaching people Te Reo Māori. And so this one is actually talking about you're more likely to get the mower back from your neighbour to mow the lawn than actually bring back a mower. And so that is really it. I hope you guys have enjoyed it. I'm happy to answer any questions whatsoever, and I will stop sharing the screen.
Kia ora. Thank you so much, Nick. That was fascinating. And yeah, you covered a lot of ground. I have so many questions, but I'm going to open the floor up to the room. If you've got a question for Nick, please either pop it in the chat now, or you can use the raise your hand function. We'll be able to see that, and you can unmute yourself and ask Nick directly. So while you guys are getting sorted, Nick, I'll jump in. And obviously the genetic troubles that you covered, particularly in terms of very small population sizes, having those real big issues. I wondered if you could comment, just for interest. I know I recently came back from Chatham Island, where obviously there was a big good news story of the black robins, which are now 445 from, I think, 5 breeding pairs. So can those issues be got around in that instance of something that's alive with a really good, genetic thing? Or can you sort of clarify what the difference is there?
So you can, in populations of endangered species like the black robin, is you could sequence ancient genomes of black robin work out by comparing it to the living descendants of Old Blue. And I remember my grandparents' house had a big painting of Old Blue in their lounge. Is you can reintroduce all that lost genetic variation back into um, uh, genetically engineered black robins, for example, and then introduce those into the population. There will be issues with any of that. So if you are trying to get these genetic variants into populations and have them stay in the population, you need large populations with short generation times that are fast breeding. And so that what we call genetic drift, which is the random chance that a genetically engineered lineage will be fixed in the population or lost in large populations, quite rare. In small populations, which characterize a lot of our endangered species, that they're small, low numbers, slow breeding, long lived, breeding with their relatives, that random chance of loss or fixation is really, really high. So there will be challenges, but it's a much wider conversation that we need to have as a society. So if we think of the moa, is the conversation to bring back the giant moa should be across all of Ngāi Tahu, all 8 iwi at the top of Te Ihu, across New Zealand. And we know that Ngāi Tahu Runanga said that the local Runanga Ngāi Tahu in Christchurch had the rangatiratanga the authority to sequence a moa genome from their rohi or tribal area for a bone found there, but not a bone from any other area. And to bring back a moa or a genetic engineering, that is a Ngāi Tahu-wide decision. And the majority of all the tribal councils and Runanga and Whānau are all dead set against it. So this technology is going to require a lot more wider engagement than, say, what we do for research projects where we're trying to engage as locally as humanly possible.
Kia ora, thanks, Nick. Anybody else got a question for Nick? You've stunned them all into silence.
Nick, I just wonder if there's any way of reconciling with Colossal. Is there any hope that they can appreciate your arguments and see the pitfalls?
Bluntly, no. They have called us a small band of angry critics that do not represent the overwhelming positivity they see around the world to bringing back moa. It's interesting in New Zealand, there's not much positivity for it, and they tend to call us armchair scientists. So they're very dogged in that what they're doing is they have a duty to do this. But for example, the moa de-extinction isn't about conservation. As Peter Jackson wants to bring back moa, he has tens of millions of dollars. And the danger, I think, is if ultimately Colossal and Canterbury Museum and the Ngāi Tahu Research Centre get a no from Ngāi Tahu, is I just think Peter Jackson and Colossal will actually just go somewhere else and cherry pick support to bring back moa somewhere else in New Zealand. Colossal doesn't value scientific criticism. They don't like academics, and Ben Lam's been quoted in stuff saying they talk to governments and politicians and regulatory bodies and indigenous people and sometimes academics who are experts in their field. And so that quote should tell you everything about what they think of us.
Thanks, Nick. I've got a follow-up question on that note with Colossal. Just with my science communicator hat on and as a devil's advocate, if I was trying to develop a new technology, and I wanted, public support and funding to develop it, even if I was true blue conservationist and wanted to do it for conservation purposes, something like the dire wolf, which is obviously a huge brand in popular culture, particularly people in my generation, is a really good flagship project to drum up, obviously, and it's worked. Like, a huge amount of public interest in things. But then it would be using that, and then, as you say, turning it into conservation, which clearly is not the priority for them. If they'd had a different approach, could you see it going differently? Could you see it have what would you change about the way it's being done, if anything, to make it a worthwhile endeavor, or an appropriate endeavor?
I think it comes down to, it's a hard question, and I'm not sure I've got an easy answer. I think it comes down to a failure of science communication by scientists. And so Andy Pask has said that he's got more funding out of Colossal signing up to de-extinct the thylacine to develop these tools to be used for real conservation than he's ever got before. So, like, effectively make a deal with the devil to get the funding that you need. And the direwolf is interesting because bringing them back, there's no conservation angle to it. Their ecosystem disappeared 13,000 years ago. So I think as scientists, we really need to go, how do we actually communicate why we need these tools for conservation? And I think if we can work out how to communicate all of that, then we could start getting funding from the private sector and philanthropists and celebrities, rather than saying we're going to go de-extinct these animals for these supposed reasons that, when you start drilling down into it, actually all fall apart. It's a hard one that I don't have an answer for. It's like those, if you're ever having interviews for funding of, like, why should Joe Bloggs, the taxi driver, the checkout operator, care? And they can be really quite hard ones to answer. But I think we need to work out how to communicate more effectively.
Cool. Thank you very much. We've got a couple minutes. We've probably got time for one more question before we wrap up, just on 8 o'clock this evening. If anyone's been sitting on something you're burning to ask, now's your chance.
I've just got a comment that I live in Wairarapa, and so just down the road from Peter Jackson. And a year or two ago, I approached Peter to fund a fossil kit for the House of Science series of useful fossil kits to raise awareness of paleontology, and because it's such a good basic inroad, a way to get people enthused about science. Never heard back from him.
Yeah, I'm not surprised. And the interesting thing is, we've done a lot of engagement with Ngāti Kahungunu Ki Wairarapa, which will be your local hapu, and they're very much dead set against de-extinction. It's like, we know that Colossal and their collaborators have been doing excavations in the Wairarapa, and private moa bone collection. Now, the interesting thing is paleontology is, like, if we are doing science, we cannot touch private collections. That's effectively in the paleontology rulebook. Yet Colossal is sampling a whole lot of stuff from Peter Jackson's private collection to do a whole lot of science on. So it's a very, very different way of operating, and quite a very different way of doing science communication as well, because you're dealing with a big it is multinational company now.
Thank you both. Ilmaz has got his hand raised. This will be the last question. Ilmaz, go ahead.
You're on mute, sorry.
Yep, thank you. Sorry, my apologies. Kia ora, Nick. Thank you for your great talk. And to be honest, I probably was stunned into silence, having followed the headlines in the news, and I really appreciate you bringing the complexity and the kind of, I guess, machinations behind this by what is obviously very well funded company. But just to give us a sense of perspective, to the best of your ability, assuming this would work, they jump through all the hoops, and what sort of price tag are we talking about, and how that might compare, for instance, to a year's funding for DOC, just to, give us a sense of how kind of much money is getting thrown at this?
I don't know what DOC's yearly budget is, but it's a good question. But Bean Lamb and Colossal did a Q&A event with the movie director of the latest Jurassic World movie to talk about real de-extinction science. And in that movie, the guy with all the money basically said it costed about $75 million to bring back one extinct animal. And Bean Lamb was asked, what do you think about that? He says, yeah, it's in the right ballpark. So $75 million for every extinct animal. Let's say you got 3 dire wolves. That's 70, 140, 210, $235 million for 3 dire wolves. So given the hundreds of millions of dollars it's going to cost to do these things, I very much doubt, say, Colossal will actually genetically engineer and create enough Frankenmoa to have a sizable, genetically secure population that they won't be completely inbred.
And of course, if there are, like you alluded to, if there are complications like all these things, there are the potential costs of that will be socialized. So yeah, thank you very much for sharing your story, Nick. That's really appreciated.
Oh, no worries.
Thank you, Nick, and thank you, Ilmaz, and thank you all for coming. We've just rolled over 8 o'clock and you're all still here, so it shows how interested everyone is in this discussion. But we'll wrap it up there. We have been recording this session, and so once we've had a chance to sort of top and tail the recording, that will be uploaded to our YouTube channel, where you'll be able to find it, and I'll send out a link to everyone who registered to come to the talk today. This is one in a series of talks, so if any of you are interested in giving a talk, please contact your respective special interest group convener, whether that be the outreach group, the paleontology group, or the geoethics group. We'd love to hear from you as well. But for now, thank you so much, Nick, for your time this evening and for sharing your insights and concerns. And it's given us a lot to think about. But also made us all more informed going forward for this ongoing debate. And I'll be interested to see how it plays out in the next few months to years. So thanks again, Nick. Thank you all for coming. Take care and have a good night all.
No worries. And if anyone wants me to give this talk to their classes or departmental seminars, or if if anyone else would be interested, just flick me an email. More than happy to.
Wonderful. Thanks so much, Nick. Take care, everyone.